Hi everyone, this lecture is going to be part 2 of your chapter 9 from the Merib text, which is covering muscles and muscle tissues. Okay, so in part 1, we finished talking about all of the anatomy of our muscles. So mostly that anatomy was microscopic, looking at the different types of connective tissue layers, the different sarcoplasmic reticulum, T-tubules, looking at the neuromuscular junction. Okay, so this second part of the lecture is now going to be looking at physiology. So what's actually happening on a cellular level during a muscle contraction, okay? And again, I mentioned this in the last lecture, but we're going to be looking at specifically skeletal muscle. Your book gives some information and differences about cardiac muscle versus smooth muscle, but we aren't going to have enough time to cover that, so we're just going to stick to talking about skeletal muscle. Okay, so the big picture, what we're going to be covering for physiology, we can divide the muscle contraction into 4 steps. So the first is the events at the neuromuscular junction. Then we can talk about muscle fiber excitation, contraction, coupling, and then cross-bridge cycling. So we're going to be talking a lot about some different anatomical structures and features of the muscle. So it's really important that you have a good basis of understanding of these different anatomical features. So if you haven't watched part 1 yet, or are still struggling with that, I'd recommend going through, rewriting your notes, organizing, drawing everything out to make sure that you really understand the anatomical features before you start diving into the physiology. Okay, so this is like the first chapter that's like really heavy on the physiology, and a lot of little steps and things you need to remember. So it's really important, again, that you have that anatomy down. Okay, so let's start first by talking about the events at the neuromuscular junction, which is my favorite part. I love this stuff. Okay, so I put those 4 steps up here in the corner so you can kind of organize your notes and make sure you keep track of where we're at in this process. So at the neuromuscular junction, first thing that has to happen is an action potential. AP stands for action potential, arrives at your axon terminal. Okay, so remember, action potential is the signal coming down from your central nervous system down the axon of your motor neuron, and it reaches the axon terminal, which is that little bulge at the end of the motor neuron. Okay. Once the action potential reaches the axon terminal, voltage-gated calcium channels open. Calcium is going to enter the motor neuron. Okay. Remember very quickly about in chapter 2 or 3, I forget what chapter it is now, I talked about at rest, the ion concentrations, and you thought of yourself on an island. All you have on your island is a banana, right? So I'm going to draw it out here for you. All you have in your island is a banana. So this is my island. I have a banana. Outside, we have salt water, right? And like a little coconut or something. I don't know. Coconut. Okay. So banana on the island. That means on your island, in your cell, at rest, you have potassium. Outside, salt water. Sodium and chloride is in salt. And in our coconut milk, remember I said we had calcium in that coconut milk. Okay? So if you understand, again, remember these concentration differences at rest, it'll help you understand as we're opening ion channels what's going to happen. Okay, so we talked about here, if calcium channels open, calcium enters the motor neuron. Okay, which makes sense because if you look here, inside your island, inside the cell, you have only potassium. So if we open a voltage-gated channel, which just opens due to the depolarization of the action potential, that's allowing calcium to move. If you know at rest calcium is outside of the cell, then the only way calcium is going to want to move is into the cell. Follow that concentration gradient. Okay, it's going to diffuse passively just move down its concentration gradient and go into the cell. Okay, so in all the ion movements we're talking about, if you know where the ion is at rest, it's really easy to figure out if a channel opens and allows potassium to move, you know it's going to want to move down its concentration gradient. Okay, so in this case, calcium channels open, calcium enters the neuron. Okay, calcium is really important at the neuromuscular junction because calcium triggers the release of the acetylcholine neurotransmitter from the motor neuron into the synapse. Okay, acetylcholine diffuses across the synapse and binds to acetylcholine receptors on the sarcolemma. Remember, the sarcolemma is on the muscle. It's the outside the cell membrane of the muscle. Acetylcholine receptors are sodium chemical gates. Okay, so if acetylcholine binds to the sodium chemical gates, it opens those gates. What way is sodium gonna want to go? You know, at rest, sodium is outside the cell, so sodium is gonna want to go into the cell. Okay, so acetylcholine binds, opens the gates. Sodium enters the muscle cell, results in what we call an end plate potential. Okay, this also we can refer to as depolarization. Okay, after acetylcholine does its thing as depolarizing the muscle cell with sodium, we don't want acetylcholine to stick around in the synapse because it's going to keep activating that muscle. Okay, and we don't want that because we only want the muscle activated when we have want the contractions to occur. So we have an enzyme called acetylcholinesterase, which is an enzyme. Right? If it ends in an A's, it's normally an enzyme that's going to just break down acetylcholine so it doesn't keep binding. Okay? So that's really what's happening at the neuromuscular junction. Okay, and I have pictures to show all of these steps. Okay, so we have, again, our motor neuron coming down, synapsing at the neuromuscular junction onto a muscle fiber. Okay, so the first thing that happens is we have the action potential, AP, coming down and reaching the axon terminal of our motor neuron. Okay, and the action potential comes down from the central nervous system, the brain and spinal cord, comes down the axon and reaches the axon terminal of our motor neuron. Okay, and that again is at the neuromuscular junction. After the action potential comes down, what it's going to do is depolarize. Okay, so the action potential depolarizes the inside of the cell here. Okay, because remember, at rest we have a negative 90 millivolt resting membrane potential. We're not going to talk about it until a later chapter, but just trust me that the action potential is going to depolarize that. So it's going to make the inside more positive with the use of some ions. Okay, that depolarization is going to cause voltage-gated calcium channels, step 2 here, to open. So we can see here we have voltage-gated calcium channels on the axon terminal. Those channels open due to the depolarization of the action potential. Calcium enters the cell. Again, at rest, you have greater concentration of calcium outside. So calcium enters the axon terminal. Calcium entering is what allows our synaptic vesicles filled with the neurotransmitter acetylcholine to be released. So there's going to go through exocytosis, and the neurotransmitter acetylcholine is going to be released into the synaptic cleft. Remember, the synaptic cleft is just the space between the axon terminal and the muscle fiber. Okay. Acetylcholine is going to diffuse across the synaptic cleft after being released via exocytosis and bind to acetylcholine, we call them cholinergic. Think acetylcholine, cholinergic ligand or chemical gated channels on the sarcolemma, right? So I'm zooming. On the sarcolemma, we have these ligand-gated channels. So acetylcholine diffuses is going to bind to these ligand-gated channels and cause them to open. Okay? What happens when those channels open is you have the movement of sodium and a little bit of potassium, but sodium is the most important thing. So acetylcholine binds to the ligand-gated channels, the channel opens. The most important thing here is sodium is going to enter the cell, okay? Because at rest, remember, sodium has a greater concentration outside. So if a sodium channel opens, sodium is going to follow its concentration gradient and go inside of the cell. Okay? Sodium enters, if you read here, it produces a local change in the membrane potential, and this is called the end plate potential, also known as depolarization. Okay, you have a positive sodium ion entering, remember at rest we have a negative 90 millivolt inside. It's going to depolarize the area here, make the inside more positive than it normally is. Okay, that's called the end plate potential or depolarization. Okay, and then the last thing that happens is, again, we don't want excess acetylcholine in the synapse. So we have the enzyme called acetylcholinesterase that's going to break down acetylcholine to make sure it doesn't continue to bind. That makes these ligand-gated channels close so that sodium and potassium aren't able to move. Okay? So, from the top, an action potential comes down the axon into the axon terminal of your muscle or motor, motor neuron. Okay. The action potential coming down is going to open calcium channels, and calcium is going to enter the axon terminal. Calcium causes these vesicles filled with the neurotransmitter acetylcholine to bind and exocytose the neurotransmitter acetylcholine into the synapse. Acetylcholine is going to diffuse across the synapse and bind to ligand-gated receptors on the muscle's sarcolemma, right? And the sarcolemma, again, is just the cell membrane of the muscle. That binding of acetylcholine to these channels is going to open them. The main important thing that's going to happen is we have sodium is going to go into the cell. That positive charge going into a negative environment, remember resting membrane potential, is going to cause an end plate potential or depolarization. So depolarization or end plate potential. Okay, and then the very last step here is, again, we don't want too much acetylcholine just floating around the synapse. So an enzyme, acetylcholinesterase, is going to break apart acetylcholine into inactive parts so it can't continue to excite the muscle. Okay, so you should be able to walk me through step by step exactly what's happening at the neuromuscular junction like that. Okay, I would recommend you take time and draw this out or print out an image like this and walk through, again, step by step to make sure you understand every single thing that's happening at the neuromuscular junction. Okay, so we just talked about the events at the neuromuscular junction, right? Action potential comes down, calcium comes in, acetylcholine's released, acetylcholine binds to the receptors on the sarcolemma, and that causes sodium to come into the muscle cell and create an end plate potential or depolarization. Okay, so that's the first step of causing muscles to contract. We then have muscle fiber excitation. Okay, so that's step 2 that we're going to talk about. So muscle fiber excitation. So the sarcolemma is going to end up becoming polarized, right? Meaning that there is a voltage across the membrane. Okay. And that is due to that end plate potential caused by sodium coming into the muscle due to acetylcholine binding to those receptors. Oh, this is saying, I didn't read the whole thing. The resting sarcolemma is polarized, meaning there's a voltage across the voltage across the membrane, resting membrane potential, negative compared to outside, so negative 90 millivolts is that resting membrane potential. Okay, the muscle fiber excitation is this end plate potential that we just talked about. Okay, so electrical changes causes action potentials in the muscle cell itself. Okay, and this is going to occur in 3 steps. So we talked about number 1. I just jumped ahead and talked about it as well. The end plate potential. Then we have depolarization and repolarization. Okay, so let's talk about that first. So first, again, remember we have at rest the resting membrane potential of negative 90 millivolts. Okay, and we talked about the depolarization due to sodium entering. So if you, again, add a positive charge to a negative space, you're going to become more positive. Okay, and this more positive, this is called depolarization. Okay, we'll also see that we have something called repolarization. So if you polarize again, so in repolarization, we have potassium leaving. If you're losing positive charge, you're going to become more negative. So you're going to repolarize that cell and get it back to rest. Okay, so we'll talk about these steps in detail, but I just wanted to start by showing depolarization, you're bringing in more positive. Repolarization, you're making it more negative again. Okay, so we stopped by talking about the end plate potential. So the end plate potential happens when acetylcholine is released from the motor neuron and it binds to the receptors on the sarcolemma. These chemically gated or ligand, I keep saying, is the same thing as chemical. Chemically gated or ligand gated ion channels open. Sodium goes into the muscle fiber. Some potassium leaves, but not much. That sodium coming in causes the inside to become more positive or less negative. And this is what results in that end plate potential or depolarization. Okay, so again, I'm going to zoom in here. When the acetylcholine binds, we have, most importantly, a positive ion sodium coming in. It's going to cause a positive area on the inside, and that's what the end plate potential or that local depolarization is. Okay. That local depolarization is going to cause the propagation of an action potential within the muscle cell. Okay, and we call that depolarization throughout the muscle cell. Okay, so we have a local depolarization due to those chemically gated channels. If there's enough depolarization there are voltage gated sodium channels in the nearby membrane that are going to respond to that end plate potential. Okay, so say in this image, our neuromuscular junctions over here. So we have the end plate potential, a positive charge over here. If that positive charge is big enough to reach over to these voltage-gated sodium channels, they are going to open due to that change in voltage from the end plate potential. Okay, so what happens is that depolarization can move down. So what happens is that voltage change due to the end plate potential causes voltage-gated sodium channels to open. Sodium is going to rush into the cell, which is going to cause more positive charge. Okay, and that's going to keep moving. There's going to be another voltage-gated channel down here that's going to sense this change in voltage. It's going to open, allow sodium to come in. Sodium comes in, and the same thing's going to happen, and we're going to call that propagation. Is where you have this movement of depolarization moving along as one voltage-gated channel opens, causing sodium to come in. The next voltage-gated channel opens, causing more to come in. And you're going to have that movement continuing down the whole length of the sarcolemma. Okay, and this leads to what we call an action potential, which is just that movement of these ions that is unstoppable and ultimately leads to a muscle fiber contractions. Okay, so that action potential is going to move again along the sarcolemma or the cell membrane of your muscle, one voltage-gated sodium channel at a time. So it's going to start at one, cause depolarization. That depolarization is going to spread, cause another voltage-gated channel to open, spread, so on and so on and so on, until it moves all the way down to the next area that's important. Okay. As that's happening, the muscle cell wants to reset its resting conditions. Okay, so we have those sodium channels doing their thing, propagating the action potential along. But we want repolarization to happen. So that's getting that membrane back to its resting membrane potential so that the inside has negative 90 millivolt potential. Okay, so as the sodium voltage-gated channels close, we're going to see voltage-gated potassium channels are going to open. When voltage-gated potassium channels open, potassium is going to follow its concentration gradient and leave the cell. If you have a positive ion, potassium is positive, leaving the cell, the inside is going to end up being negative again, right? Because you're causing positive ions to leave. Okay, and that's going to restore that resting membrane potential where the inside is more negative than the outside. And it does this to make sure that the other voltage-gated channels don't just continue to stay open because there's positive inside. We don't want a positive inside all the time. We want to make sure that we lose that depolarization of that positive and those positive ions leave so that we don't trigger those voltage-gated sodium channels to allow more sodium in. And that's called repolarization. So we have a refractory period, which is the time that the muscle fiber cannot be stimulated until this repolarization is complete. Okay, and that's important because we want the muscles to relax between contractions so that they don't become fatigued or you don't, cramp up. So after repolarization, obviously now we have potassium would be on the outside and sodium's on the inside, which is not where they're found at rest. Remember, potassium's in bananas, that's inside the cell on your island. Sodium's in salt, that's in the salt water outside of your island. So you're going to have sodium-potassium pumps actively, this is an active process, pumping the sodium potassium in and out of the cell to restore that resting membrane potential and that resting separation of concentration. Okay, and we talked about sodium potassium pumps before, how they work. So they use energy to pump sodium potassium back to where they belong. Okay, so that's what's happening to excite the muscle fiber. Okay, so back to this chart. Now we should really understand what's happening. So we have the cell at rest, negative 90 millivolts. Okay, just a reminder. Inside the cell at rest, you have potassium. Outside the cell at rest, you have sodium, calcium, and chloride. So this here is showing the action potential in the muscle fiber. So the first thing that really happens is we have the end plate potential, which again happens just at that neuromuscular junction. Okay, the acetylcholine is allowing some sodium to enter the cell. That's going to depolarize it, but it's depolarizing it enough just to allow the voltage gated channels to do their thing. Okay, so the end plate potential uses ligand gated channels. They depolarize it enough so that the voltage gated channels can work. Okay, so the first step we have is depolarization, which we have sodium channels, sodium ions going into the cell, depolarizing it, make the inside more positive. The sodium channels are going to close, and they're going to have repolarization happening. In the repolarization, we have potassium channels open, and potassium goes out of the cell. It leaves. If you're losing a positive, you're going to become more negative, and you're repolarizing again. At the end, potassium channels close. And another thing I want to kind of add to this chart is that after all that happens, you're going to need the sodium-potassium pump to restore that separation of concentration with sodium-potassium. Okay, so that's what's happening in the muscle fiber. So the muscle fiber is becoming excited. Okay, the third step, we had excitation, contraction, coupling. And this is pretty straightforward. It's just the events that transmit the action potential along the sarcolemma, which we just talked about is the excitation part of it. That's going to be coupled with the sliding of myofilaments, the contraction part of the muscle. Okay, so now we're kind of combining that action potential, the excitation of the muscle, with what's going to allow those myofilaments to actually contract. So the action potential goes all the way along the sarcolemma until it reaches those structures that are so important that we talked about earlier, the T tubules. So that action potential goes down the T tubules. In the T tubules, you have voltage-sensitive proteins that ultimately are going to cause the release of calcium from the sarcoplasmic reticulum. That's important because calcium leads to contraction. Okay, so the action potential just causes calcium to be released, and that's going to what's going to lead to the contraction. But the action potential ends before the contraction actually happens. Okay, so let's show this in a picture. Okay, so this is blurry, but let's zoom in. So we talked about action potential coming down causes calcium to go into the cell. Right, causes the vesicles with the neurotransmitter acetylcholine to be released, diffuse across the synaptic cleft, bind to acetylcholine receptors, cause sodium to enter the muscle, which results in the end plate potential. So that's the events at the neuromuscular junction. Then you're going to have propagation of that action potential, right, where you have now voltage-gated sodium channels, allowing sodium in, depolarizing it. I guess I should say this. Sodium coming in is going to depolarize. We have repolarization, with potassium channels allowing potassium to leave. Again, these are all going to be voltage gated channels. That's going to propagate all the way along until it reaches the T tubule of the sarcolemma. And remember, the T tubule is just an inversion of the sarcolemma. So that depolarization is going to go down the T tubule. And what that depolarization going down the T tubule is going to do is initiate the release of calcium. And if you remember from when we talked about the anatomy, calcium is stored in the sarcoplasmic reticulum. Sarcoplasmic reticulum is where it stores calcium. Also in the terminal cisternae, right? Those are the perpendicular parts that make up the triad with the T tubule. So when the depolarization comes down the T tubule, it initiates the release of calcium from the sarcoplasmic reticulum and the terminal cisternae. Okay. So the next slide kind of shows that a little bit better. So I'm going to zoom in. Action potential propagates down, again, goes into the T tubule. That depolarization down the T tubule allows these channels, we're not going to really talk about the physiology of those, but ultimately it's going to allow calcium that is stored in the terminal cisternae of the sarcoplasmic reticulum to be released into the muscle, into the cytosol of the muscle. Okay? And that is going to couple with the contraction. So calcium is what's really important in allowing cross bridges, which is step 4. The Crossbridge cycle is step 4. The cross bridges to form. Okay, so what calcium does, calcium comes in. If you remember, troponin and tropomyosin. Tropomyosin blocks the active sites on actin, the thin filament. Okay, remember we want our myosin heads, the thick filament, to bind with the active sites on actin, the thin filament. Tropomyosin at rest blocks those active sites, so you can't have cross bridges forming. What calcium does, calcium comes in here, binds to troponin. You need to know calcium binds to troponin. So calcium binds to troponin. What that does is causes a conformational change, and it moves tropomyosin out of the way so that the active sites on actin are exposed. So after calcium binds, now you see that the tropomyosin is out of the way, and the myosin head now can bind to the active sites on the actin. And that's what forms a cross bridge. Okay? So some words for that. So if we don't have calcium released, we don't have intracellular calcium, tropomyosin blocks the active sites on actin. Myosin heads cannot attach. The muscle remains relaxed. Okay, so that is if we don't have calcium. There's low intracellular calcium. When T tubules become depolarized, these voltage-sensitive proteins cause the release of calcium from the sarcoplasmic reticulum. So this calcium enters the cytosol inside of the muscle. When we have high intracellular calcium concentrations, calcium binds to troponin. Troponin changes shape and moves tropomyosin out of the way. Okay, so troponin isn't the one blocking the actin active sites. Tropomyosin is what's responsible for blocking those. Okay, but when calcium binds to troponin, it changes shape and moves tropomyosin out of the way so that the myosin heads can bind to actin, which forms that cross bridge. Okay, that initiates the cross-bridge cycling, which ultimately leads to sarcomere shortening and muscle contraction. When the nervous stimulation ceases, calcium is pumped back into the sarcoplasmic reticulum and contraction ends. Okay, so if we have any sort of calcium present in the cytosol, it's going to be bound to troponin. Tropomyosin is going to be out of the way so that the active site on actin is exposed and there will be cross bridges formed. Okay, so if calcium is present at all, that's going to happen. So when we want the muscle to relax, we have to pump all of the calcium out of the muscle so that it unbinds to troponin, and tropomyosin goes back and covers those active sites. Okay. So that's excitation, contraction, coupling, right? The last step of a muscle contraction is the actual cross-bridge cycle, which is the actual shortening, the sliding filament theory with actin and myosin, the cross bridges forming, pulling against each other, and shortening the length of the muscle. So this only has 4 steps. So we have the cross bridge formation. The high energy myosin head attaches to the actin thin filament active site. Step 2 is the power stroke. The myosin head pivots and pulls the thin filament towards the M line, which is in the middle of your sarcomere. Cross bridge detachment. ATP attaches to the myosin head, which causes the cross bridge to detach. And then step 4 is the cocking of the myosin head. So ATP is hydrolyzed, and the energy from that cocks back the myosin head into a high energy state, getting it ready for its next attachment. Okay, so then you go back to the top again. If the myosin head can attach, you form a cross bridge. There's a power stroke where that head shoots forward and pulls the thin filament. It detaches, and then you cock the myosin head back to get it ready for another power stroke. Okay, and that's the cross bridge cycle. So here's a picture of it. So the first thing, again, you have the cross bridge formation. The myosin head binds to the active site on the actin. The power stroke, that head pulls, right? Pulls and moves the actin along, which is going to cause the shortening of the sarcomere, right? The cross bridge is going to detach. So ATP attaches to the myosin head and breaks that bond between actin and myosin. Again, you need ATP for the detachment. ATP is hydrolyzed. So that means it's converted to ADP in a phosphate group. And that takes the actin or the myosin head from this like low position to a cocked position, ready to again attach, then do a power stroke, move it this way, detach, and then cock again. Okay, so that's the whole step-by-step process of a muscle contraction. Okay, so what you should be able to do is go through step by step. So at the neuromuscular junction, what's happening? You have an action potential coming down that causes calcium to go into the axon terminal, calcium allows the synaptic vesicles to release acetylcholine into the synapse. That's going to attach to ligand or chemically gated channels on the muscle cell. It's going to allow sodium into the muscle, which is going to cause the end plate potential. After the end plate potential, that causes a local depolarization, which is going to activate voltage-gated sodium channels. Those are going to open. Sodium is going to go into the muscle cell, causing depolarization, which is going to propagate along. After that, you're going to have voltage-gated potassium channels open. Potassium leaves, which repolarizes, which is going to also lead to the sodium-potassium pump, right? Getting back to resting membrane potential. Okay, but this depolarization is going to propagate. So depolarization is going to propagate along the sarcolemma until it reaches the T-tubule. It enters the T-tubule and it allows calcium to be released from the terminal cisternae of the sarcoplasmic reticulum. Okay, calcium goes down, binds to troponin, tropomyosin moves. This allows the myosin head to attach to actin active sites, forming the cross bridge. Okay. After the cross bridge is formed, you have the Crossbridge cycle. So you form the cross bridge, you have the power stroke. ATP causes the actin and myosin to detach. ATP is hydrolyzed, causing that myosin head to cock and get ready for another Crossbridge cycle. Okay, so this is going to keep happening so long as there is calcium in the muscle cell. Okay, so we want to use energy to pump calcium back into the sarcoplasmic reticulum to allow the muscle to relax. Okay, the only way the muscle is going to relax is if calcium is pumped back. Okay, so you should be able to go through this step by step and tell me exactly what's happening where. Okay. I'm sure everyone's heard of rigor mortis, which happens about 3 to 4 hours after death when your muscles begin to stiffen. This happens because you don't have ATP being synthesized. So calcium cannot be pumped back into the sarcoplasmic reticulum. Okay? Remember, we need calcium in the sarcoplasmic reticulum for the muscles to relax. If we don't have ATP to pump calcium back into the sarcoplasmic reticulum, those cross bridges will continue to form, the muscles will continue to contract, and you don't have any muscle relaxation. You also need ATP for crossbridge detachment. So it leads to this constant state of contraction at maximum contraction. Okay, so rigor mortis is what happens when muscles stay contracted until the muscle proteins finally break down and cause those muscles to be released. Okay. And there's one more thing I want to talk about, which is the effect of botox. Which for this, I'm going to go back to the neuromuscular junction. So I'll go here. Okay, so let's talk about botox. Botox is a type of toxin. It's actually botulism toxin that they use in very small amounts, cosmetically most of the time, to reduce wrinkles. But they can also use it to treat different tension headaches. You can use it to stop hyperhidrosis, which is excessive sweating. And it, for cosmetic purposes, reduces wrinkles because it stops muscles from contracting. Okay? And it does this by working at the neuromuscular junction to stop certain events. Okay? So what Botox does, this toxin is injected into your muscles. Okay? And what it does is it stops these vesicles from fusing with the membrane in the axon terminal. So acetylcholine can't be released. So this doesn't happen when Botox is there because it stops the release of acetylcholine. Okay, so Botox, if it stops the release of acetylcholine from these synaptic vesicles, you never have the end plate potential. You don't have depolarization, so you can't ultimately get calcium to be released from the sarcoplasmic reticulum, so you can't have the tropomyosin moved out of the way so actin and myosin can bind. Okay, so it stops this whole process at the neuromuscular junction by stopping acetylcholine from being released. Okay, so that's what Botox is doing. A lot of people think it's like the similar thing to fillers, but fillers are actually injecting kind of a jelly-like substance in the skin to add some definition. So if you see someone with lip fillers, that's a very different thing than Botox. Botox is stopping muscles from contracting, which is reducing the appearances of lines and wrinkles that are caused by muscles contracting. Okay, and Botox, when they inject it, takes about 3 days to start working for that Botox to get in and stop acetylcholine from being released. And it lasts only about 6 months. So the effects of Botox are reversible. So you're not going to get Botox, and if it, it's going to paralyze your face forever. Slowly, acetylcholine will start being released again, and you'll return to normal functioning. Okay, but that's what Botox does, and I think that's pretty cool. Okay. There's still about 20 slides left in this, so I'm going to stop right here and then continue the rest of this chapter talking about whole muscle contraction and muscle twitches in a part 3, just to break this up a little bit. Okay, so there will be a part 3.